So, glycine and proline are generally thought of as helix and beta sheet structure breakers, but they're often found at the beginning and end of helices and sheets. They're very often found in terns. Glycine, because of its ability to adopt conformations restricted for other amino acids, and proline, because of its rigid phi angle of -60 to -77 degrees. So if we look at proline and its isomers, we see that um, it can exist in the trans configuration and in the cis configuration. Again, this is going to be referring to um, the presence there of that carbonyl, either in trans um, to the nitrogen atom in the ring, or in cis to it. Remember, 99.6% of peptide bonds for proline are in the trans conformation, and about 6% of peptide bonds in the cis configuration, and many of these are found in beta terns. That leads us to supersecondary structures, also known as motifs, and these are recognizable combinations of alpha helices, beta strands, and loops that occur in many proteins. Some examples of these motifs are helix loop helix, which literally looks like a helix with a loop with a helix. Coiled coil motifs, helix bundle motifs. Um, the beta alpha beta unit, the hairpins, the beta meander conformation, the Greek key, and the sandwich conformations. That brings us to tertiary structure. Now, tertiary structure refers to the entire three-dimensional arrangement or conformation of a protein chain in 3 dimensions. So again, it refers to the overall 3D structure of individual polypeptides. Tertiary structure often results in the formation of domains, which are discrete, independently folded regions of a protein, often compact units that have some sort of function. And so here you can see we have our secondary structures, but the overall representation is of a tertiary structure, where now we've got secondary structures linked together in 3 dimensions, and each of these separate domains has some kind of purpose within the protein. It might be an enzyme activity, it might be an interaction activity with another protein or with another molecule like DNA, but the domain is going to have some discrete function. It's going to be an independent region into and of itself, a compact region that's going to give the protein some kind of unique activity. And so this portion of the tertiary structure has been labeled as a domain, and of course there's another domain here that you can see as well on the right side. There is structural similarity between lactate and malate dehydrogenase. And so in the picture below, we have Bacillus stereothermophilus lactate dehydrogenase. And in picture B, we have E. coli malate dehydrogenase. And as you can see, they look very similar in terms of their structure. But indeed, they're different proteins. They're in the same family. Their structures are very similar, but they're only 23% identical. So these are called homologs. Different proteins from the same family but only 23% identical. The picture on the right depicts that of the enzyme pyruvate dehydrogenase. And what you can see is there are many types of secondary structures that have now wrapped together into this tertiary structure. Um, and within this tertiary structure of pyruvate dehydrogenase, we have several types of protein domains. We can first talk about protein domains generically, what kinds of structures are made up by protein domains. And we could talk more specifically like our textbook does, and which I'll get to in a little while. So if we look at pyruvate dehydrogenase, you can see we have an area where we have a lot of beta sheets. We have areas where we have primarily alpha helices, or we have mixtures of the two. And so on one level, we can talk about protein domains as in terms of their secondary structure. So we have all alpha, which would consist entirely of alpha helices and turns, also known as loops. And if we look here in the middle portion of pyruvate dehydrogenase, we see a lot of alpha helices. We have protein domains which are all beta, and you can see at the top portion here of pyruvate dehydrogenase, a lot of beta sheets coming together to form a functional domain within the protein. We have alpha/beta domains where you have mixtures of the two structures interspersed amongst each other. For example, a beta sheet going into an alpha helix going back into a beta strand, right? So a beta strand going to alpha helix going back into a beta strand, and you can see that down at the bottom portion of pyruvate dehydrogenase. And then there are alpha plus beta protein domains where you have a mixture of each of these structures with local clusters of each as well. And so each type of secondary structure arises from a contiguous region of the polypeptide. And if you look at the bottom portion, particularly the bottom right portion, you can see a mixture of the two structures of alpha and beta um, um, clustering together. And so these designations of all alpha, all beta, and so on refer to the secondary structures within the protein domains. But remember, the domain is a tertiary structure that's a consequence of those secondary domains being arranged in three dimensions and giving a unique property to the protein. And so our textbook talks about some specific types of protein domains. One of those domains is called a TIM domain or a TIM domain, which is a donut-shaped domain, where the inner portion is composed of a beta sheet and the outer portion is composed of alpha helices. It talks to you about other types of domains, such as the SH2 domain or the PTB domain. The SH2 domain was first found in the SRC virus, also known as the sarcoma virus, and that's where it gets its name, SH, for Circ homology. Domain stands for phosphotyrosine binding domain. Your book talks a little bit about the NAD binding domain, which serves as an important redox transfer cofactor interactor. And so there's lots of types of protein domains that have been identified, and each of them has a particular role within the protein. The designations on this slide, 1 through 4, are really referring to the secondary structures within the protein domains. And as we move forward, you may hear me make reference to certain specific types of protein domains, like our textbook does. Like, for example, the TIM domain or the SH2 domain, or even your textbook talks about something called the EF-HAN domain, which is a domain that selectively binds to calcium ions. And so that's a domain you'll hear us talk about again as well in the textbook. So protein domains are very um, um, important components of proteins, which give proteins their ability to do their said functions. These are some of the interactions that maintain tertiary structure. As you can see, there are interactions such as hydrophobic interactions, salt bridges that are ionic in nature, water bridges, and hydrogen bonds, as well as disulfide bridges. So these are the kinds of interactions that are going to help contribute to tertiary structure. So thus far, we've talked a lot about secondary structure. Recently now talking about how a lot of secondary structures linked together and folding in upon itself form tertiary structures. Well, here are some interactions very important for tertiary structures. The disulfide bridges, the hydrophobic interactions, salt bridges, water bridges, and hydrogen bonding. Now, quaternary structure is when you have an arrangement of two or more polypeptide chains into a multi-subunit molecule. We have organization and arrangement of subunits in a protein, and each subunit is a separate polypeptide. So a quaternary structure is when you have the interaction of these separate polypeptides, these subunits, into an overall multimer or oligomer of protein. And that multimer can be a dimer or a trimer or even higher quaternary structure. Proteins with only one polypeptide component are called monomers, and the subunits may be identical or they may be different polypeptides. They're usually held together by relatively weak forces such as hydrophobic interactions and van der Waals forces. And as you can see here, we have an example of a quaternary structure. This um, variable domains of the IgG molecule. And so we can see we have the antigen binding sites, the FC region. We have some um, disulfide bonds holding this together and forming a quaternary structure. Other examples of quaternary proteins are numerous. A handful of such examples shown here. Various antibodies or lysozyme, an important protein in immune system defense. So molecular shapes or conformations of proteins impact protein function. These quaternary structures can assemble via protein-protein interactions. This table shows the natural occurrence of various oligomeric proteins in Escherichia coli, the prokaryotes. And the first column talks to the different types of oligomers observed, from monomers, dimers, and trimers, all the way down to dodecamers and higher oligomers and very long polymers. And what you can see here, first of all, there's about 372 proteins in E. coli, which would be considered oligomeric, where you have a quaternary structure coming together, multiple subunits. And so within this, we have monomers forming oligomers, accounting for about 19% of all of the oligomeric proteins observed in E. coli. Dimers coming together, accounting for about 38% of the types of oligomeric proteins that are observed in E. coli. And by far, dimers are the most common of the oligomeric proteins, quaternary structure course, if you go down the list, you can see that there are some um, types of higher oligomeric structures that also participate in this quaternary structure. So within E. coli, almost 400 proteins exist as oligomeric structures where you have different polypeptides coming together to form quaternary structures that are needed for function. So this is an example of some of the big protein complexes that are observed in a prokaryotic cell like E. coli. Starting with the leftmost area, we have pyruvate dehydrogenase structural core, which is a homomultimer with many subunits. Many ribosomes found throughout the cytoplasm of the E. coli needed for protein synthesis. RNA polymerase, a multi-protein complex needed to make RNA from DNA. Groel, a chaperone protein, many copies of that protein as it assembles into a homomultimer of at least 14 monomers. So these are just some examples. Could have picked any number of examples to show you these macro protein complexes and how they are assembled um, from the biochemical point of view. So these large macromolecular complexes uh, can exist as a single protein present multiple times, like Groel, can contain more than just protein. For example, the ribosome contains protein and RNA, and some of these complexes can be very large. So the picture you see at the top of this slide would depict that of an enzyme called hexokinase. And that's kind of the gray, bluish looking structure in the space filling model. And around that enzyme, you see molecules of water. And this talks to the hydration of proteins. So protein surfaces will be hydrated in an aqueous environment, and the interactions with water impact the protein's function and the protein structure formation. So again, if you look at the top of this slide, you see the hexokinase along with hydration, water surrounding it. And if you look in the upper right, you'll see actually like a separate structure distinguished from the rest of the protein. And that's actually glucose. And as you can see, glucose is also hydrated. And it happens to be the substrate for this enzyme hexokinase. A kinase is an enzyme that phosphorylates something. What's being phosphorylated here, a hexose sugar called glucose. And so that hexokinase is going to have to accept the glucose into its active site. And in fact, in order for that to happen, the glucose has to actually shed the water. And in the slide that's going to come up here below, in the picture below, you'll see the glucose has entered into the active site of the enzyme. And in doing so, it has shed its water. So this hydration of proteins is not a trivial thing because active site of the protein is often going to have to be free of the water, although not always. But there needs to be a way for the hydration around the proteins to move if necessary. And so here in the description of what has happened, we see the hexokinase enzyme, and its normal job is to phosphorylate glucose. And so as that hydrated glucose above enters the binding site in a cleft in the enzyme of hexokinase, it actually loses those water molecules and enters the active site. And that water exclusion process actually helps facilitate the conformational change that's needed to open up the catalytic domains and allow the enzyme to act upon the substrate in this instance. And so the exclusion of water is an important part of this enzyme's action. And it also prevents unproductive hydrolysis of ATP, which is also needed for the phosphorylation to occur by this enzyme. So the moral of the story isn't to necessarily go into all of the details of hexokinase, but it is to show you that proteins are hydrated just like other molecules in solution, and that hydration is an important aspect of proteins in the cell, because cell cytoplasm is water. That leads us to the next topic within proteins, which is protein-protein and protein-ligand interactions. In the realm of interpolypeptide interactions, and we'll call these protein interactions, we have various forces at work. We have non-covalent interactions that can mediate these interpolypeptide interactions, including hydrogen bonding, van der Waals forces, London dispersion forces, charge-charge interactions, among these. There can be covalent bonds that cause interpolypeptide disulfide bridges. The subunits of multi-subunit protein complexes are held together so tightly inside cells that they rarely dissociate inside cells. And when it comes to the association, for example, of two polypeptides, we can depict this mathematically as polypeptide 1 and polypeptide 2 coming together to form an interaction of P1, P2, indicated by that semicolon. And we could actually calculate rates of interaction or rates of association. So there's a forward rate called the association rate and a reverse rate called the dissociation rate. So at equilibrium, we basically see that the rate, the forward rate, is proportional to the concentration of both of the polypeptides, and the reverse rate is going to be proportional to the two polypeptides which are now interacting, and then at equilibrium these two rates are going to be equal. And so K1 over K minus 1 will equal the concentration of the two polypeptides together divided by the concentration of each one separately. And we can call that the Ka for the association constant. And this is not to be confused with the acid dissociation constant of things we talked about earlier in the course. In biochemistry, it's generally done to discuss protein-protein interactions in terms of dissociation events. So when it comes to the dissociation of two polypeptides, there we have the combined dimer falling apart into each of the individual components. We have a forward rate of dissociation and a reverse rate of dissociation. And so at equilibrium, these rates are proportional to the concentration of each of the species. And we can calculate Kd for the dissociation of two polypeptides as concentration of each of the separate members divided by the concentration of the interacting polypeptides. So that's the Kd value. And it's called the dissociation constant. Similarly, for protein ligands, which is abbreviated here as PL, we can have the protein plus a ligand. What is a ligand? A ligand is any chemical that binds to a receptor, and here the receptor is a protein. So P plus L forms a protein ligand complex, and the protein ligand complex can be looked at in terms of dissociation, just like we did for the protein-protein complex above. So here, Kd is going to equal the concentration of the protein times the concentration of the ligand divided by the concentration of the ligand bound to the protein. So Kd is going to talk to us about the dissociation of ligand from the protein. And so if we look and we compare the Kd to the Ka, what we find is that Kd is equal to 1 over the Ka. Now, a low dissociation constant means tighter binding. So if we start to compare the binding of ligands to their proteins and look at dissociation constants there in the third column, what we see is that the lower the dissociation constant, the tighter the binding. And so if you look at biotin binding to avidin, it has a tremendously low Kd. That's a very strong interaction. Or insulin binding to its receptor, it has a very low Kd, so very tight binding of ligands to the protein. Comparatively speaking, if you look at calcium binding to calmodulin, a calcium binding protein, it has a much higher Kd value than, for example, biotin binding to avidin. And so if you look at the next portion of the diagram, you have Kd in the middle. High affinity binding would be in the area of 10 to the minus 10 to 10 to the minus 16 molar. Low affinity binding would be in the area of 10 to the minus 2 to 10 to the minus 6 molar. And the color bars here indicate the range of dissociation constants which are typical for various classes of interactions in biological systems. So if you look at the olive green bar, that tends to be the dissociation constant for antigens binding to antibodies, or the other way, antibodies binding to antigens. Or if we look at the blue bar, it's typically the Kd range for ligands binding to receptors. And if we look at the pink bar, it's typically the range of substrates binding to enzymes. And so a few interactions, such as that between the protein avidin and the enzyme cofactor biotin, fall outside the normal range. The avidin-biotin interaction is so tight that it's generally considered irreversible. Sequence-specific protein DNA interactions reflect proteins that bind to a particular sequence of nucleotides in DNA, and that's as opposed to being just a non-specific binding to any DNA site. You can have sequence-specific protein DNA interactions, and that becomes important in particularly in gene transcription. So this concept of Kd is very important. Again, it's inversely related to the Ka. So the smaller the Kd value, the tighter the binding of the ligand for its protein, and that talks to you about affinity. So if you have a low Kd, you have a very high affinity. So we've introduced some words here like dissociation, ligand binding, and affinity. So feel comfortable understanding this relationship as we move forward. Particularly, it's going to have relevance in pharmacology again when we talk about these topics there. In the next part of this lecture, we're going to talk a little bit about three-dimensional protein structures denaturation and renaturation, and Anfinsen's thermodynamic hypothesis. The native conformation of a protein can be disrupted by chemical changes in the environment or by addition of heat. So proteins are sensitive to such kinds of changes. This is particularly relevant because many new drugs that are being developed are protein drugs and can be affected by changes in heat or environmental conditions. These changes can cause denaturation of a protein, and oftentimes only a small amount of energy is needed to denature a protein, sometimes just enough to denature 3 or 4 hydrogen bonds. Some proteins, when they denature, unfold completely and they lose all their native structure, assuming a random coil conformation and a completely random conformation. However, most denatured proteins retain some level of internal structure. For example, some of the alpha helices may still be maintained despite denaturation. Generally, the strength of the structure inversely correlates with the level of structure. So quaternary is less strong than tertiary, which is less strong than secondary, which is less strong than primary in terms of strength and ignoring all disulfide bonds. The graph on the right shows the heat denaturation of ribonuclease A. And in this experiment, a solution of ribonuclease A was put into 0.02 molar of KCl at pH 2.1 and was heated. And the unfolding of the protein was monitored by changes in UV absorbance, which are the blue circles, viscosity, which are the red circles, and optical rotation, which are the green circles. Relatively small range of temperature unfolding is a cooperative process, so destabilization of a few weak interactions can lead to unfolding or loss of the entire native conformation. And you can see that here, pretty much starting at about 23 degrees Celsius, small changes in temperature cause big changes in destabilization and denaturation of the protein. Most proteins have a characteristic melting point called a Tm, which is dependent on pH and on ionic strength. So you might be surprised to see that this protein is significantly denaturing at 37 degrees Celsius, which is what it would normally be found at in the body. But again, the conditions by which this experiment was done was in a solution of potassium chloride at 0.02 molar and at a very acidic pH of 2.1. That brings us to the topic of chemical denaturants, of which there are several. The first of these are chaotropic agents, and chaotropic agents include things like detergents and organic solvents, and these are compounds which destabilize protein structures. They are the opposite of cosmotropes, which would stabilize protein structures. Chaotropes destabilize protein structures. And the way they go about this is through decreasing hydrophobic interactions, or disrupting ionic interactions, or disrupting hydrogen bonds in proteins. Some of the chaotropes are depicted for you here on the right. As you can see, we have urea and guanidinium chloride. And both of these chaotropes are effective disruptors of hydrogen bonds. Of course, pH is a chaotrope in the regard that it can disrupt ionic bonds as you change pHs, and detergents can act as disruptors of hydrophobic bonds, um, as can some of the organic solvents like butanol, for example. So there are several types of chaotropes. Two of them are depicted here on the right, urea and guanidinium chloride, which both are hydrogen bond disruptors. In the class of chemical denaturants, there's also reducing agents, and these are things like 2-mercaptoethanol, and sometimes that's called beta-mercaptoethanol, and dithiothreatol, sometimes that's called Cleland's reagent. And what these do is they reduce disulfide bonds in proteins. So if you have a disulfide bond occurring between two cysteines in a peptide, 2-mercaptoethanol or dithiothreatol will reduce that disulfide bond and leave behind free sulfhydryls on the protein structure. And so, as I mentioned, there are other environmental factors as well, like pH. Extreme pH can completely denature proteins by disrupting ionic bonds, and varying the ionic strength of the solution can also act as a denaturant. So, among the chemical denaturants, we have things like chaotropes, which would include things like urea and guanidinium chloride, um, would include detergents and other organic solvents, reducing agents like beta-mercaptoethanol or dithiothreatol, and pH and ionic strength. All of these can serve as chemical denaturants of proteins. So if we look, for example, at cysteine here, and we have two molecules of cysteine coming together, they can react to form cysteine, or I should say cysteine, like Christine. Remember, a dimer of cysteine residues. And so in proteins, we can get disulfide bridges that form. In the peptide below here on the left, you see these yellow structures which depict disulfide bridges. So the arrows are pointing out to you that, for example, there's a disulfide bridge between cysteine 26 and cysteine 84. So one molecule of cysteine forming a bridge there. A disulfide bridge between cysteine 40 and cysteine 95, for example. And so disulfide bridges are critically important for holding tertiary and quaternary structures together. One protein that you may be very familiar with, insulin, has several disulfide bridges which contribute to its tertiary structure. So these disulfide bridges are very important, and reducing agents, like we saw on the last slide, like beta-mercaptoethanol or dithiothreatol, they will disrupt this disulfide bond, uh, converting the cysteine back to um, free cysteine residues. Within ribonuclease A, which is a very well-studied enzyme, there are several disulfide bonds. As you can see here, there are several cysteine residues, as pointed out by the yellow C in the amino acid sequence here that you see before you. And the disulfide bonds in ribonuclease A um, are occurring between the blue link cysteines here. So here we've got cysteine, let's see, 66 with cysteine 121, and those which are being pointed to by the red arrows, so cysteine 84 along with cysteine 136. Okay, those are also forming disulfide bonds. So those disulfide bonds are very important for the folding of ribonuclease A, which, if improperly folded, cannot function to cleave RNA. Okay, so these disulfide bonds contribute greatly to the three-dimensional or the tertiary structure of the enzyme, and oftentimes in situations where we have quaternary structure, where we have polypeptides coming together, disulfide bonds may be involved with the quaternary structure as well. Okay, so the other arrows here are pointing to other disulfide bonds that are found in the ribonuclease A enzyme. And therefore, denaturation of ribonuclease A requires reducing agents. Right? In order to denature this enzyme, um, the presence of a reducing agent like beta-mercaptoethanol, for example, is required. A very important concept worth discussing at this point has become known as Anfinsen's postulate, and it's really based on thermodynamics. In the 1950s, Anfinsen proposed that the information determining the tertiary structure of a protein resides in the chemistry of its amino acid sequence. We kind of take that for granted now, but that was initially proposed back in the 1950s. And here, Anfinsen was studying the reversible denaturation of proteins following cleavage of disulfide bonds and disruption of tertiary structure. And what he found is that there are many proteins, but not all, but many that could spontaneously refold to their native form. And so this led to a thermodynamic hypothesis that he put forward, which said, quote, the three-dimensional structure of a native protein in its normal physiological milieu, meaning its normal solvent, its normal pH, its normal ionic strength, the presence of other compounds such as metal ions or prosthetic groups, the normal temperature, and any other factors that would normally be going on, is the one in which the Gibbs free energy of the whole system is lowest. That is, the native conformation is determined by the totality of interatomic interactions and hence by the amino acid sequence in a given environment. And this was a big deal. This wasn't something that was well understood at the time, and has been found to be um, highly applicable to proteins. Much of the initial investigations that Anfinsen carried out were performed on ribonuclease A, the enzyme we just discussed, that possesses multiple disulfide bonds, and he would study how that ribonuclease folded. In fact, the Nobel Prize in Chemistry went to him for this discovery in 1972. And so if we look at the native ribonuclease A, and we see it's got several disulfide bonds. And what Anfinsen found is that if you denature this with urea and 2-mercaptoethanol, you can reduce all those disulfide bonds and have what's called the denatured form of ribonuclease A, which is no longer active and able to cleave RNA. Now, if you remove the mercaptoethanol but leave the urea, you'd have oxidation in the presence of urea. So the sulfhydryl groups could pair randomly, but there'd be no hydrogen bonding. And this led to inactive ribonuclease A with randomly formed disulfide bonds, and so wasn't able to have its enzymatic activity. Only about 1% of the protein forms correctly to recover activity under these kinds of conditions. Now, there's 8 sulfhydryl groups with 105 possible pairings, and there's only one conformation that's correct. So, if he now removed the denaturant and kept reducing conditions intact, so if he removed the urea and left just a little bit of mercaptoethanol, he could reconstitute the native ribonuclease A, which would be active. And so this was very interesting. The reason why we think it worked is that by leaving little amounts of mercaptoethanol, just trace amounts, those misformed disulfide bonds could be reduced and have a chance to reform again. And in fact, this is what Anfinsen found. And so, removal of the of the denaturant and very slightly reducing conditions allowed reshuffling of the sulfhydryl bonds. And this allowed for activity to be regenerated. The conclusions from Anfinsen's experiments with ribonuclease A were that the correct disulfide bonds form only after formation of the native conformation, that renaturation of ribonuclease A is spontaneous and driven entirely by the free energy gained in changing to that stable physiological conformation, and that the conformation of ribonuclease A is determined by the primary structure. And so this was a large discovery during the day. It led to a great understanding of how amino acid sequence can predict structure. It's far from perfect. There are examples where complex folding occurs, um, but it was a very large advancement in the field of structural biology, which led to Dr. Anfinsen actually winning the Nobel Prize in the 1970s. And so here's a picture I found of him working in his lab and looking very motivated to understand the folding of proteins.